Hello and welcome to our lecture 11 on population genetics, specifically Hardy-Weinberg population genetics. I'm sorry for throwing the syllabus a little bit out of order, but this one works a little bit better in the online scenario. If when you go through this lesson you have any questions or are having trouble with any of the concepts, please do not hesitate to send me a message in the Remind app. So the first thing to go over is what exactly are we going to talk about when it comes to Hardy-Weinberg law, and why are we doing this lesson? Now, the first question that I pose to you on this slide is, how does Hardy-Weinberg law or Hardy-Weinberg equilibrium lesson differ from what we studied so far? And the key here is that we previously focused on individual genetics crosses. So for instance, you cross this Apterus fly with this wild type fly, what's going to happen? But if we're using DNA for purposes like forensics or ecology, what we call the everyday life examples, well, you need to then look at a population as a whole rather than just individuals. Okay, and the other point here is when we say looking at the population, we then get into population genetics. And what we use this for is basically we end up using Hardy-Weinberg laws to explore relationships between allele frequencies and genotype frequencies in an ideal population. And when we say ideal, that's where the idea of equilibrium comes into play. Okay, so I'm going to write the notes on the screen that we just went over verbally. And whenever I write HW, that's not homework, that's Hardy-Weinberg. Okay, so now when you use Hardy-Weinberg in order to study population genetics and in order to explore that relationship between allele frequencies and genotype frequencies, now before we get into the two predictions that help us do this, I want to point out, remind yourself that allele means the variations of a gene. So for instance, a capital A would be one type of allele, a dominant allele, whereas a lowercase a would be a recessive form of an allele. Whereas genotype frequencies, when we talk about genotypes, we talk about two capital letters representing a homozygous dominant genotype, the heterozygous hybrid, which is a capital and a lowercase allele, and the homozygous recessive. Okay, so there are two alleles and there are three genotypes we're going to focus on. Now, when we do these Hardy-Weinberg calculations and population genetic studies, Hardy-Weinberg makes two predictions. The first one is that the frequencies of the alleles in a gene pool do not change over time. So basically what that means, there should be no disturbing effects like mutation or selection or migration or anything that would change the frequency of dominant versus recessive alleles. Okay, so our first prediction is frequencies of the alleles in a gene pool do not change over time. And remember, this is referring to, Hardy-Weinberg is referring to an ideal population, a population at equilibrium. So it makes sense that you don't want to see frequencies fluctuating in something that's supposed to be stable or at equilibrium. The second prediction is that if two alleles are at a locus, okay, so for instance, each locus we already know has two alleles because one came from mom, one came from dad. Then after a generation of random mating, the frequencies of your genotypes, meaning homozygous dominant, heterozygous, and homozygous recessive in a population, can be calculated by the Hardy-Weinberg equation. Okay, so first I'm going to write down what I just said, and then we'll take a look at the actual equation. So if two alleles are at a locus, okay, and the alleles can either be dominant or recessive, then after a generation of random mating, okay, it's got to be random mating, then the frequencies of the genotypes, again, there should be three genotypes. There's homozygous dominant, there's our heterozygote, and then there's homozygous recessive. So you can calculate them. With the Hardy-Weinberg equation. Okay, and the equation is p squared plus 2pq plus q squared should equal to 1. And there are a few things that you have to understand when you look at this equation. Okay? The first thing you have to realize is that when you see the letter P in this equation, okay, that letter P is representing the frequency of allele dominant. Okay, so in this case we can say P is the frequency of allele capital A. Okay, then when you see the letter Q here and here, Q is the frequency of the lowercase, the recessive allele. Okay? Now, let me just erase the screen so that you can really focus in on this concept. Okay? If you have this equation, p squared plus 2pq plus q squared equals 1, right? And we just said that p equals frequency of allele A, and Q equals frequency of the recessive allele, right? Well, then P plus Q must equal to 1. Because think about it, in math, 1, remember when we talked about probability and p-values, 1 is the same as saying 100%. And as you know, when you're looking at a genotype or the different alleles for a genotype, you only have 2 possible alleles per trait. Okay, you have that dominant allele, you have the recessive allele. So both of those, the frequency of having dominant and recessive, that dominant plus recessive has to equal to 1 because there's no other allele option here. Okay, and when you look at this crazy, scary looking equation, p squared plus 2pq plus q squared equals 1, well, here, let me change the color so that you can really think about what this means. Think of a Punnett square, right? If we're saying p squared, right, and we said p is the frequency of capital A, or your dominant allele, what does p squared then mean? P squared means capital A times capital A, right? Which you know from your Punnett square, when we have 2 heterozygotes crossed, you have that one doubly dominant genotype possible. Then 2pq. Well, if we said P is capital A and Q is lowercase a, then what we're saying here is 2 capital A lowercase a, right? Well, where have you seen 2 capital A lowercase a? Well, let's go back to our Punnett square. Capital A, lowercase a, capital A, lowercase a. Look, 2 of the heterozygous genotype, right? And then what's left? There's one more left, q squared. That's the third genotype. That's the recessive, fully recessive one. And what do you know from our little Punnett square? You have one square that's double lowercase a. Do you see any other squares other than dominant, homozygous, homozygous recessive, and the hybrids? No. So all of those have to add up to 1, right? 100% of the possible genotypes in the cross. Okay? So that's why you see p squared plus 2pq plus q squared equals 1. P squared is homozygous, dominant, homozygous, dominant. Sorry, that was very sloppy handwriting. Then we have our second one is the hybrid genotype. And our third one is the homozygous recessive. Okay, so don't get confused when you see these letters and different values. Just keep in mind that ultimately all of your genotypes possible for that cross should result in 1, because that they're representing 100% of the possible genotypes. And P plus Q should equal to 1 because there are only 2 possible alleles. Okay, so in your notes, make sure you circle, star, highlight this equation the p squared plus 2pq plus q squared equals 1, and this one here, p plus q equals 1, because these two will guide all of the work that you're going to do for Hardy-Weinberg, okay? And this slide that we just did, the purpose of it was for you to have background information and understand, you know, why does p squared plus 2pq plus q squared equals 1? What do these random letters actually mean, okay? Now you'll also hear it said that when the two alleles do result in the predicted genotype frequencies, the population is said to be in Hardy-Weinberg equilibrium. Okay, and we're going to go through in the upcoming slides what that actually means, you know, when your population is in equilibrium and why it's in equilibrium. And if the numbers, the genotypes and alleles are out of whack, then what does that tell you about the population? Okay, so we're going to go through some examples to see how exactly we use Hardy-Weinberg and what it all means. Okay, so the first thing that we're going to do is use Hardy-Weinberg equilibrium and equation laws in order to calculate genotype frequencies in a population. So the example that I could give you is I'll tell you that a population is in equilibrium. And we usually write it as if the population is in equilibrium. Okay, and I'll give you frequencies of both alleles, or I could even give you the frequency of just one of the alleles, and automatically you should be able to know the frequency of the other. So for instance, I could say if in equilibrium and the frequency of the dominant allele, capital A, is equal to 0.7. Okay, what's the first thing you can figure out once I've given you that information? Well, right away, you can figure out the frequency of lowercase a, right? Because if capital, the dominant, is 0.7, what does the lowercase have to be? 0.3, right? Because the frequency of the dominant plus the frequency of the recessive have to equal 1. So if I give you the frequency of one allele, you can automatically figure out the other by simply doing 1 minus the frequency of the first allele. Okay, and that gave me 0.3. Then, with that information, I could figure out the frequency of each of the genotypes, right? So we start with homozygous recessive. And, you know, ask yourself what these letters actually mean. Well, the probability that you get from mom, a capital A is 0.7, right? Let's say the first allele. Then you have a second allele because you got one allele from each parent. So if parent number 1 gave you the capital A with a frequency of 0.7, well then, what's the probability of your second allele that's capital? Well, that's also 0.7, right? Because we said for this equation, the frequency of capital A is always 0.7. So the probability that that second allele has a capital A is 0.7 and was acting up. Okay, 0.7. Okay, so that tells you that the probability of a zygote having that genotype, capital A, in that population would be, so probability, capital A, well, that would be 0.7 times 0.7, which is equal to 0.49, or you can say 49%. So that's basically, this calculation we just did basically tells you 49, acting up, 49% of the time. Genotype, capital A, or homozygous dominant, will occur in this population. Okay? Now we could have done that simply using the p squared plus 2pq plus q squared equals 1. Okay, so you know AA is the same as saying p squared, so 0.7 squared is equal to 0.49. Okay, so you didn't have to write all of this out that I just did, but I wanted you to know the background behind this. Then you have capital A, lowercase a. So you look at the probability of getting capital A from the male parent, from dad, okay, with lowercase a from the female parent. Well, that probability is 0.7 for capital A, right, times 0.3 for lowercase a. And I'll erase those just so that you don't think I'm dividing by anything in your notes, but that was to show you what the 0.7 and 0.3 were for, okay? Now, is that the only way that you can get a heterozygote? No, you can also get a heterozygote by getting the dominant allele from mom, okay? So there's a chance of getting dominant allele from mom and getting recessive from dad. So that's also that is 0.3 times 0.7. Or in this case, 0.7 times 0.3 again. And that's why in the equation that we saw earlier, it's the same as saying 2pq, 2 times the allele, dominant allele, times the recessive allele, okay? So 2 times the dominant, which is 0.7, times the recessive, which is 0.3, okay? So when you do that, 2 times 0.7 times 0.3, that gives you 0.42 or 42%. 42% chance of getting capital A, lowercase a, the heterozygote in this population. Then the last thing to do is to get the frequency of the homozygous recessive. And that one would simply be, what's our frequency of lowercase a? 0.3 times 0.3. Okay, 0.3 times 0.3. Again, you can think of it as q squared. 0.3 squared is the same thing as saying 0.3 times 0.3. And that gives you 0.9 or 9%. Okay, probability of getting the homozygous recessive in this population. Okay, so make sure you can put a square around each of the percentages. So 42% chance for the genotype that's heterozygous, and the last one to put a square around is over here, and that is the fully dominant. Okay, I'll put a little star next to each of those values because those are your final answers. Okay, so notice in the beginning, all I gave you was the frequency of a single allele, the dominant allele. And I could have given you either one. I could have given you the recessive allele instead. Doesn't matter. When you are given a single allele for a population that's in equilibrium, you can then calculate so much. You can calculate the second allele's frequency in that population. You can calculate the homozygous dominant genotype frequency in that population, the heterozygous frequency in that population, and the homozygous recessive frequency in that population. So from one little number, 0.7 frequency of capital A, you got everything else that is written on this slide. If you have any questions on how to use this to go from an allele frequency to calculate genotype frequencies, just send me a message and remind, and I'll give you some more examples, and I'll go over this slide again. So now, what if instead of being given the allele frequency, what if you're given the genotype frequencies for a particular population? Okay, so in this example, I'm going to show you two different ways to tackle a problem in which you're given the genotypes and need to figure out the allele frequencies. So in this example, we're going to have a population of 100 individuals. Now, keep in mind, because we're talking about genotypes and alleles, if you have 100 individuals, each of those people have 2 alleles for the particular trait that we're looking at, because you get 1 allele from mom and 1 allele from dad. So keep in mind, 100 individuals means 200 alleles. Okay. So I'm going to write out the frequencies of the 3 different genotypes. So we have 79 out of 100 people in this population have the genotype 1, 1. Okay, and this is actually a real genotype I'm going to use for this example. So out of 20 out of 100 have the genotype called 1, delta 32, and only 1 out of 100 has the genotype delta 32, delta 32. Now this is a very important genotype in the world, okay, because this genotype, delta 32, meaning you have 2 of the, we'll call it the recessive allele, since before we were looking at capital A and lowercase a. So 2 of the lowercase allele, delta 32, people with this are resistant to HIV, because it basically causes a mutation in the receptor of your cells that HIV usually binds to and recognizes and uses as like an entry, a door of entry into your cells. These people with delta 32 genotype, they have a mutated form of that receptor. And so HIV cannot properly recognize and bind and infect those people's cells. If you're heterozygous for it, you won't be fully resistant to HIV, but HIV in these people may progress slower. And then you have the first one. Unfortunately, most of the world population is 1, 1. So when we're looking at this problem, allele capital A is 1, and allele lowercase a is delta 32. So those are the names of the alleles. And if you're 1, 1 genotype, unfortunately, you can get HIV. You are susceptible to HIV, and most of the world population, unfortunately, is genotype 1, 1. Now, the first way to tackle this problem, you've been given 3 genotype frequencies, okay, 79 out of 100, 20 out of 100, and 1 out of 100. Now, if you want to know the allele frequencies, the first way that you can do this, option number 1, I'll write it so you remember in your notes. Option number 1, to calculate allele frequencies of this population. Okay, what you can do, I'll change the color, is simply count the alleles. Okay, so what that means is we have two alleles. We want to know their frequency. Well, our first allele is allele that's named number 1. We can count everywhere that we find it in our genotypes. So where do you find allele 1? Here, here, and here. You find it 3 places. That first one that I circled, what's the frequency of that? 79 out of 100. So my pen is acting up. 79 for that first one. For the second place that we found it, what's that frequency? That's also 79. And then what's the third frequency? 20. Okay, that gives us 178 alleles, are number 1, out of the 200 possible alleles. So 178 out of 200, that's the same as saying 178 over 200, which is equal to 0.89 or 89%. Okay, so that would give you, trying to see where to write it so we don't lose it, 0.89 or 89% of this population is the number 1 allele. Then I'll change color so that it doesn't get lost in the mix. The second allele that we have is delta 32. That's our second allele. Where do you see delta 32? That first spot is 20 individuals. Okay, so there's 20 delta 32 alleles there. Where's the next spot that you see delta 32? Right here. And that's one allele. And the last spot that you see it here, well, that's 1 allele again. So that's 22 out of 200 alleles. Okay, and that's 0.11 or 11% of the population, which makes sense because before we said the first allele was 89% of the population, which means the second allele has to be 11% frequency in that population because the two alleles have to, their frequency, if it's in equilibrium, has to equal to 100%. There's no other allele for that trait in that population. Okay? If you're unsure of how to get allele frequencies by counting the alleles, simply message me in my mind and I'll go over it again. The second way that you can get, sorry, I'm just getting my pen ready. The second way that you can get allele frequencies is you can go by genotype frequencies. Now we said there are 3 genotypes in this example. There's the 1-1, the 1 delta 32, and the delta 32, sorry, let me erase that, the delta 32, delta 32 genotype. The first genotype, 1-1, we said that the frequency was 79 out of 100 individuals. Well, mathematically, what frequency is that? 79 out of 100 means 79 divided by 100, right? 79 divided by 100, which is equal to 0.79 frequency. The second genotype, we said, was 20 people out of 100 had that one. So you do 20 divided by 100, which gives you 0.20 frequency. And the third one, we said 1 out of 100. So you would do 1 divided by 100, which is 0.01 frequency. Okay? So now that you wrote out your genotype frequencies, okay, that's what you were just writing out, genotype frequencies. There are your 3 genotype frequencies. Now you want to figure out the frequencies of your alleles, right? So based on that, the frequency of allele 1, is, where do you find allele 1? Right here in the first genotype, 0.79 frequency. It is that allele 1 represents that whole genotype, right? It's 1, 1, which means there's no other allele there, so that entire 0.79 represents allele number 1. Then, where's the next genotype that you see allele number 1? The second genotype. But what do you notice about it? Allele number half. Only half of that. Okay? Sorry, my pen is acting up. So it's only half of that. So you now say plus 1/2 of the frequency of the heterozygote, which is 0.2. Okay? That gives you 0.89 or 89%, which we already knew from our previous calculation. 89% is the frequency of allele 1. Okay, and for your notes, in case you forget where we got the numbers, 0.79 is the frequency of homozygous dominant, and then 0.2 is the frequency of heterozygous. Okay? And then the last thing to calculate is the frequency of our second allele, which is delta 32. Where do you see delta 32? Well, the first place we see it is the other half of the heterozygote genotype. So 1/2 of that 0.2, the heterozygous, plus the entire homozygous recessive, which is 0.01. Okay, and what does that give you? Well, half of 0.2 plus 0.01 gives you 0.11 or 11%, which we already said was the frequency of delta 32. Okay, so 11% is the frequency of delta 32 in this population. And for in your notes, in case you forget what it means, 89% is the frequency of allele 1 in this population. Okay? If you have any questions or concerns, you're not sure how to do this, just let me know in the Remind app. You're going to get to practice some in the post lab, so hopefully it'll become a little clearer when you're doing this out on your own. But what I recommend is for each of these slides that I give you an example. Once you have watched the video and taken your notes down on each slide, go back to these examples, go to the original question I asked before I started drawing all of the answer out, and see if you can calculate it. You know, from these same numbers, if someone gave you the genotype frequencies of 79, 20, and 1, would you be able to calculate the allele frequencies? Okay? Let me know if you have any questions or concerns. Now, the last thing for us to do is to really use this Hardy-Weinberg as robustly as we can Now that you are experts at calculating the genotype frequencies of a population, the allele frequencies of that population, well, now we're going to use all that information and Hardy-Weinberg to test for equilibrium, to test whether or not a population is actually in equilibrium. And I'm going to show you how to do this in 3 steps. Okay, so the first thing that I'm going to do is I'm going to write exactly what the example is. So our example, I'll give you a population. This population includes 283 individuals. Okay, and I'm going to abbreviate individuals, of which you have 223 have genotype 11. Actually, I'm going to go back to the capital and lowercase because I think students get a little confused with 1 and delta 32 sometimes. So have genotype capital A, 57 have genotype capital A lowercase, so they're heterozygous, and 3 have genotype lowercase a. Okay, so remember you need to have 3 genotypes. You have your homozygous dominant, your heterozygous, and your homozygous recessive. Now, the first thing you're going to want to do when trying to figure out if this population is in Hardy-Weinberg equilibrium, the first thing you want to do is ask yourself, what are the frequencies of these genotypes? And all that means is you need to translate numbers and language into math, right? So the genotype of our frequencies means we need the frequency of capital A, the frequency of capital A, lowercase a, and the frequency of lowercase a. Well, what do you know about capital of the homozygous dominant? We said 223 out of 283 have that genotype. And what did we say before about out of in math? 223 out of 283. Whenever you hear out of, you do division. Okay, so all you do is 223 divided by the total. That gives you the frequency. Okay, that's equal to 0.788. You can write in your notes that, again, you are dividing that genotype by the total number of organisms. Okay, and I'll put it over here because sometimes if I don't write it, students don't write it. So to get the frequencies of each genotype, you do number with that genotype divided by total individuals. Okay, so then our second one, I'll change the color so you can see this, are heterozygotes. What's the frequency of heterozygotes? Well, 57 out of 283 individuals have the heterozygous genotype. So you do 57 divided by 283, which is equal to 0.201. And then we get to the very last one, our lowercase, our recessive ones. I'll use purple for this. 3 out of 283 have that lowercase genotype. Okay, so that's 3 out of 283 individuals. So the frequency is 0.011. Okay, now then keep in mind these numbers for the next slide, because now we're going to go to step 2. Okay, so I'm just going to take a second to rewrite on this slide what we just wrote in terms of the frequencies. So we had on the previous slide A frequency was 0.788. Heterozygous was 0.201. And homozygous recessive was 0.011. So now our next step, step number 2, is based on those genotype frequencies. So based on genotype frequencies that we just calculated, what are the allele frequencies? Okay, so we now want to do first the frequency of our first allele, capital A. Well, again, in order to do that, you go to your genotype frequencies and you ask yourself, where do you see the letter A, right? So I'm going to do this the second option that I showed you, where you're using the genotype frequencies themselves. So the frequency of capital A is this whole top number, this whole homozygous dominant frequency, plus, where else do we see it? The second number, but it's only half of that. Okay, our dominant allele only represents half of that genotype frequency because the other half is the recessive allele. So we say plus 1/2 of our heterozygous frequency. And that should give you 0.89 frequency of allele capital A. Okay, so we circle, star, highlight that number. That's for capital A. And then we need to calculate the frequency of our second allele, which again is half of the heterozygous, plus the whole homozygous recessive. Okay, and that should give you 0.11. Now, you also could have simply done, once you have the first number, which in this case was 0.89, you also could have just done 1 minus 0.89, because like we mentioned earlier, you have to have the frequency of both of your alleles equal to 100. So you could have done it that way as well. Then the reason why I didn't do it that way was because the point of this is to really see whether or not this frequency is in equilibrium. So since we weren't told it was in equilibrium yet, I didn't want to assume anything, so I did not want to do 1 minus, okay? So even though you technically might be tempted to do that, try to avoid it. Instead, calculate it the way we just did here, where for that second one we did 1/2 plus the homozygous genotype. The very last thing to do. Now we have the frequencies of the alleles. And again, I'm going to remind you what they are because it's not like the big chalkboard where you get to still see. The frequency of capital A we said was 0.89, and the frequency of lowercase a we calculated as 0.11. Now the last thing we're going to do is step 3, which is based on those allele frequencies. Okay, based on those allele frequencies, what should Hardy-Weinberg genotype frequencies be? Okay? Notice what we're saying. In step 1, we calculated genotype frequencies, but they weren't Hardy-Weinberg frequencies. In step 1, we calculated the observed genotype frequencies for that population, the genotype frequencies that we are seeing in the individuals of that population. Whereas Hardy-Weinberg, they want to talk about populations that are in equilibrium. So their calculations that they gave us are for equilibrium. So in this question, when I say based on those allele frequencies, what should Hardy-Weinberg genotype frequencies be? What we're going to calculate now are expected genotype frequencies. So you can go back in your notes, and where we had step 1 of this example, you can write those are your observed genotype frequencies. Now we're going to calculate the expected based on Hardy-Weinberg. And if you remember, Hardy-Weinberg said p squared 2pq and q squared. Okay, so p squared, 2pq, and q squared as the 3 genotypes. So now p squared, what's p squared? That's our first allele squared, right? So that is 0.89 squared, which if you plug that into the calculator should give you 0.792 as the genotype for equilibrium of capital A, capital A. Then we have, I'll change the color of it to PQ, and PQ, remember, that is saying capital A and lowercase a. So now we're doing 2. What's our capital A frequency? 0.89. What's our second allele's frequency? 0.11. And when you do 2 times allele, the first allele times the second allele's frequency, you get 0.196 as the genotype AA. Okay? And then our last one to calculate is Q squared. Q squared, which is just our second allele. So 0.11 squared, okay, which should give you 0.012. Genotype, double lowercase a. Now you can remind yourself underneath each of these in your notes so you remember where you got it. The first one is our first allele. In the second equation, it's 2 times first allele times second allele frequency. And then the third one is your second allele, okay? The reason why sometimes I write that out, and I'll even write up here lowercase a, that is what we mean by second allele. The reason why I write that out sometimes is a lot of times students will jot down notes in class and then they go back to read them and they're like, wait, what's this 0.11 from? What's this 0.89? What am I doing? What was I even in that class? Okay, so writing things like that out for yourself helps you then remember later on why you did it, okay? Then our last step is to actually analyze what these results just told us. So how do we analyze what the final results tell us? What does it mean if there's a difference? Well, we just calculated expected frequencies. And whenever you have expected, when we did a previous lesson way back in the past, I'm not going to say what it was because that's what you're going to tell me in a minute in the Remind app. But when we did a previous lesson, whenever you mentioned expected something, you then compared it to observed of something, right? So what we're going to do is I'm going to rewrite what we calculated on some of the other slides. Remember in step 1, so reminder of step 1 answers, which we said were our observed genotypes. Sorry, my handwriting is very sloppy today. Observed genotypes, okay? We said we have our 3 genotypes. Capital A, lowercase a, that frequency was 0.788. Heterozygous was 0.201, and the recessive was 0.011. We then, in step 3, had answers that were our expected Hardy-Weinberg genotypes. Okay, and we're going to go capital A again, heterozygous and homozygous recessive. And in our expected, we calculated 0.792, 0.196, and 0.012. So what those tell us here, these expected, these are the genotypes, frequencies, if a population is in equilibrium, if it's ideal, if it's in equilibrium, because that's what Hardy-Weinberg say. Hardy-Weinberg is for equilibrium. So what's the last step of all of this? How do we know if this population is in equilibrium? Well, we compare what we actually see in the population with what we would expect in equilibrium. So for instance, our population has 0.788 or 78.8% homozygous dominant. Okay, so I'm referring to the first one here. If it's in equilibrium, it should have 0.79.2 or 79.2% of the population as homozygous dominant. Well, those numbers are fairly close. Then we look at the second one, 0.2 versus 0.196, which when you round it is 0.2. So the expected and the observed there are fairly close. Observed 0.01, what do you notice here? Hardy-Weinberg equilibrium expected is 0.012. So when you compare those two numbers, they're all very similar. Okay, so when we look at this, it looks like expected equilibrium values are very similar to the observed. So what would that tell you? That would tell you, okay, this population seems to be in equilibrium. Now we did this by eyeballing it, which on a quiz, the final, post labs, I would be fine. That's what I'll have you do, is just eyeball it. If the numbers are close to each other, like 0.788 and 0.792, when you round them off, they're both 0.79. When you round off 0.201, 0.196, they're both 0.2. Okay? When you round off 0.011 and 0.012, they're both 0.01. Okay, 1%. So when all 3 of the genotype observed and expected are close to each other, that tells you the population is in equilibrium. If even one of them was not close to its expected, then what would we say? If there's a difference in any one of those three genotypes, you would say the population is likely not in equilibrium. Okay? So if even one of those numbers was not close to its expected, make sure to write that down. I don't have room on the screen anymore, but write that down. If even one of the genotypes is not close to its expected Hardy-Weinberg, you would say it is not in equilibrium. And usually what I do is I'll give you numbers that are very different so that you're not questioning, well, is it close, is it not? I'll give you very different. So for instance, if you're expecting 0.788, then I would give you if you're expecting 0.792, then I would give you for the observed something like 0.4, which is very different from 0.79, okay, far away from it, so that you wouldn't have to question, well, is it considered close or not? Now for the Remind question. I want you to send me in the Remind app. For this slide, we eyeballed it. We said, okay, they all round off to about the same thing. It's an equilibrium. If we wanted to test this mathematically and prove that the expected is very different from the observed, or the expected is very similar to the observed value, what technique that we did in a previous week of lab this semester could you use to mathematically prove whether or not the expected and observed were similar or significantly different from each other? Okay, send me in the Remind app what technique you would use to accurately calculate this. Okay, but like I said, in our actual class, I'm not going to make you do the further statistical analyses. Okay? Please let me know if you have questions or concerns about this. Now, if you looked at this and you saw that the expected frequencies were noticeably different from the observed frequencies, it would not only tell us that this population is not in equilibrium, but it would also tell us that one or more of the following Hardy-Weinberg assumptions are being violated in this population. Okay, so please write that down. If the expected frequencies were noticeably different from the observed frequencies, it would tell us that one or more of the following Hardy-Weinberg assumptions are being violated in this population. Okay? Okay, I know you hate the math and calculation lesson so much, but we're almost over with it. The last point that I want to make is, like I said in the previous slide, if any of your expected frequencies are very different from the observed frequencies in that population, it basically tells you that population is not at true equilibrium, and that one or more of the assumptions on this slide are basically being violated in that population. And these are the 5 assumptions that Hardy-Weinberg makes about a population in equilibrium. The first one here is that individuals of all genotypes should have equal rates of survival and equal reproductive success. Basically what that means is, if you look at the figure here, no selection would be going on in a population that's at equilibrium. So that means that, for instance, in the example that we showed earlier, one of those genotypes, is very valuable, the one that makes you resistant to HIV. And so if there was selection going on, and people only wanted to mate with people who had that very valuable genotype, then of course you're going to see fluctuations in genotypes. Eventually you might even have some of the genotypes disappearing, having, 0 frequency in that population, because the favorable ones would be so, overpowering mathematically, and you would end up with, completely being out of equilibrium. The next assumption, no new alleles are created or converted from one allele to another. What is that a fancy way of saying? If a population is at true Hardy-Weinberg equilibrium, there should be no mutations. Okay, no new alleles, because that would completely throw off these calculations. We said that P plus Q equal 1. There should only be 2 alleles. If there's now suddenly P, Q, and W, P, Q, and R, and S, that throws off our calculations. The third is that individuals should not migrate into or out of the population. Okay, because again, that would cause fluctuations in the genotype frequencies and the allele frequencies in that population, and it would not be at equilibrium anymore. The fourth assumption for Hardy-Weinberg model is that the population should be infinitely large, which you see here. Okay, infinitely large because of the fact that in practical terms, you want the population to be large enough that sampling errors and other random effects are negligible. Okay, so when we were calculating the frequencies of genotypes, for instance, or alleles, if you only have a population of four individuals, and you were mistaken about one of them because, for instance, let's say you're looking at eye color. Well, in certain lights, when the light is kind of dim, you can't really tell the difference between hazel eyes and green eyes, or hazel eyes and blue eyes. So maybe you were wrong about one of the individuals. Well, if your calculations were out of, 1,000 people, being wrong about one or two of the people, it's not going to change your numbers very much. But if there were only 4 people in your sample, and you're wrong about one of them, well, now your numbers just change drastically. So the Hardy-Weinberg model wants there to be an infinitely large population. And the last assumption is that individuals in the population mate randomly, which I know the idea of random mating, which you see over here, sounds funny, but it's basically going back to the point that we made in the first assumption. You don't want selection going on. You don't want people to be picking only one particular genotype, saying, hey, that's the best looking of the genotypes. I want to mate with that one, because that will then put everything out of equilibrium, and it will skew the numbers and the calculations. Okay, so we're not going to get into much detail beyond that, but I just want you to be aware of what it tells you when your population is not in equilibrium. The fact that one or more of these assumptions is being broken in that population. Okay, so if I gave you a problem to work out and it turns out that the observed and expected are very different from each other, you should be able to tell me, well, maybe selection's going on, maybe mutation's going on, maybe migration is going on. Maybe it was too small of a population, or maybe there was not random mating. Okay, because if the population, the observed and the expected are different, it tells you one or more of the assumptions on this page are being broken. Okay, meaning that they're not happening. Okay, contact me with any questions you have. And that is it for today. I apologize. I know it's annoying when we go back to the calculation-based lessons, but I will admit that this was a very important one because the Hardy-Weinberg concept and calculations is something that you don't just see in our class, but you see in a lot of other classes too, especially if you're then taking evolution, ecology, or environmental science courses, as well as any kind of general bio courses here or in your future. Okay, if you have any questions or having any trouble with any of the concepts today, please just send me a message in the Remind app and I can help you review them a little further. Thank you and have a great day.